So next up, we're going to explore ASHWA 30, a formulator's guide to low-dose, high-impact product innovation. Please welcome Dr. Bethapudi, who works at R&D at Natural Remedies, and her research interests include clinical and preclinical research, as well as new product developments for supplements and nutraceuticals. As we've been doing, you can ask your questions at the end. We'll get back to those, so go ahead and post those in the Q&A box, and I will turn the stage over now.
Hello, everyone. Thank you for joining today's session on this topic, a formulator's guide to low-dose ashwagandha, high-impact product innovation. Before getting into as the session started, we talked about high-impact product innovation. We will get to understand in the course of the presentation why did we call it as high-impact product innovation and how it becomes a formulator-friendly one. The whole product development started with understanding the customer unmet needs. There are multiple ashwagandha formulations in the market. So the question was, is there a need for another ashwagandha formulation? A primary market research was done before the product development started at Natural Remedies. This primary market research was supported by a reputed agency. The reputed agency has a set of questions to ask the executive level people in various dietary supplement companies. And then when we looked at the data that was mined from this primary research, there were very interesting insights from the primary market research that the market is still looking for a low dose ashwagandha. But it should be having a good efficacy with benefits on stress and endurance, with safety and regulatory compliance. Also, the other attributes with the market, which the market is looking forward for, are early onset of action, product that works, and there is no discussion and debate on the part used. It was mostly it was told that they would like to have a root formulation, and of course, sustainability being the key. So this was the starting point for this product innovation in the ashwagandha space, having a lot of insights from the consumer customers. And this is where the journey began for the low dose ashwagandha, which will be helpful for the formulators. As we all are aware that ashwagandha is not new to the industry or to the customers or the consumers. Ashwagandha is a great adaptogen, and to have a refreshing insights on ashwagandha. It's a master adaptogen. Its botanical name is Withania somnifera, and it's been used since 4,000 years in India and in Indian systems of traditional medicine. That clearly indicates how safe it is over a period of there has been a great historical use of ashwagandha. And the benefits, of course, are for endurance, stress, and fatigue. And the part used in the traditional systems of medicines is, of course, root. This is also very much known to the people that adaptogens are known for acting in the stress area. What they do, they simply help in stress protecting mechanisms and they reduce the phase of exhaustion. That's how they work on the stress related aspects. So with this background on the safety and with this background on its adaptogenic ability and with the insights from the market on the customer needs for a new ashwagandha formulation, we embarked on this product innovation. The technology that was used to develop this particular low dose ashwagandha was bot technology. Now, how is this bot technology different from the classical methods? The classical methods use concentrating the actives that may lead to toxicity or either enhancing the bioavailability of the vithanolides and altering the pharmacokinetics. But this unique ashwagandha formulation used bot technology, that is bioactive optimization technology. We will understand in the subsequent slides the interesting innovation story of this low dose ashwagandha formulation. How is this achieved? It's an intelligent combination of the fractions that work in a synergy at a low dose. So that's how this particular formulation works. Uh, so this is where we adapted the new age bot technology, and that's where Ashwa 30 got developed. Introducing to you all Ashwa 30. Why did we call it as 30? You will see more insights in the subsequent slides. So this is to be told because this is a very interesting product development insight. How the bot technology is used in this particular product development. As we understood from the traditional systems of medicine, Ayurvedic books, that it's the roots that are used. So the starting material for the product development was certainly the roots. Then a lot of leads were designed, which were from alcoholic, non-polar, and also the hydroactives. So these are the multiple extractions that were done, and there were combination of these extractions that were used. So totally, there were many leads that were developed. Now, this leads underwent an iterative investigation. Here, there are multiple mathematical models. There are mathematical models which were used. So, interestingly, in this particular product development, we used a model wherein we looked at various doses, various combination of the extracts. We got so many predictions from these mathematical models. They were again retested in biological models. It was an iterative investigation. So the mathematical models helped us in a lot of predictions. That predictions got tested in the biological models. And again, from the insights of biological models, they were put into mathematical models. So that kind of iteration, it almost took 2.5 to 3 years for the R&D team here to develop the lead, and thus the lead got finalized. And it is further optimized using the chemistry studies for temperature, pressure, and other things. And of course, the dose response and other things, it was further optimized in the biological models. And that's where Ashwa 30 was developed. So it took a lot of time for the R&D team at Natural Remedies to develop this product using this iterative investigations, optimizations, and all these things. Now, what is the phytochemistry of Ashwa 30? This is again interesting. Of course, the phytochemistry is again evolving from whatever the insights we have got during the product development. It is a dual standardization. So 30 milligram dose ashwagandha. Interestingly, this ashwagandha formulation is very effective at 30 milligram dose, the lowest dose of an ashwagandha formulation. It is standardized again to 30%. So what does this 30% ashwa bioactive complex look like? It is greater than 15% of withanolides, weight by weight HPLC, which is by modified USP method, which are very effective for the anti-stress, uh, which are very effective for a stress, and the 15% of ATP active fraction, which is very unique to Ashwa 30, is responsible for endurance enhancement. Of course, the entire product, as we looked in the product development also, it's the intelligent combination of the fractions that has a synergistic benefit. So both the fractions are very important for both the indications, that is stress and endurance. However, when we looked at the predictive transcriptomics and when we use biological models, we found that ATP active fraction is having a great benefit on the endurance. That is a precise reason why there is a dual standardization for Ashwa 30. Along with ethanolides, even ATP fraction is standardized too. And let me tell you, there is third-party validation is also done for withanolides as well as the ATP active fraction. Now, this is how the full spectrum withanolides look like. And what is this ATP active fraction? I'm sure that you must be inquisitive about the ATP active fraction. What does it contain? This is a very unique standardization. As I mentioned in the beginning of the presentation, that the dual standardization is also popping up from a lot of insights that we got during the product development. So during the product development, this particular fraction was very effective for the endurance benefits. Just to have a batch to batch consistency, the product is standardized to the entire fraction so that we don't lose on it. we have a complete understanding of this active fraction again. So the phytoactives present in it are arabinogalactons and other secondary metabolites like ferulates, saponins, and phenolics. There is definitely a batch-to-batch consistency for the entire fraction. The TLC and HPLC fingerprints are available for the entire fraction. So this is in just about ATP active fraction, which is very unique to Ashwa 30. Once the product is developed, it was again tested in the preclinical studies. In the preclinical studies, there were many models which were used. However, taking the insights from the consumers or the customers' the requirement of developing the product for endurance and the stress benefits, we looked at these three models: the stress models, the endurance models, and the anxiety models. In this, Ashwa 30 has shown 53% reduction in the corticosterone. And in the treadmill test, Ashwa 30 has shown almost 2x benefit or 105.6% benefit over the control. What does this mean? That the laboratory animals were able to run on the treadmill for a longer duration. So their endurance capacity and the energy abilities are highly improved by Ashwa 30. Anti-anxiety benefit, there was 32% reduction in the anxiety, which was noticed in the preclinical studies. So overall, the product at 30 milligram dose was able to produce anti-stress, endurance enhancement, and anti-anxiety benefits, which were very visible in the preclinical studies. It remains always important for the preclinical studies to get translated into the clinical studies. So we will be looking in the presentation how the clinical studies also showed similar results to preclinical study. And before that, this is something very interesting, a heat map which was generated with predictive transcriptomics. It's basically the gene expression, how the vithanolide molecules and how the secondary metabolites of the ATP fraction are interacting with various genes and which pathways these metabolites are targeting. So it is put in a heat map form. As we can see here, the more darker the color, the better is the effect on that pathway. For example, these are the vithanolides, and these are the secondary metabolites of the ATP fraction. As we can see here, the cellular response to stress is mostly affected or affected by the vithanolides. Vithanolides have more benefit on the cellular response to stress when compared to the ATP active fraction, while ATP active fraction has more effect on the oxidative phosphorylation pathways or the energy related pathways when compared to the vithanolides. Of course, when combined, both are synergistic. Both are very key for the for both stress and the endurance benefits. However, the way they have the effect on the genes and the pathways is quite unique. And this is one of the reason why the product is having a dual standardization again. Now, let's look into the clinical trial results, whether this product, which has shown great results in the preclinical, is translated to clinical trials. We see here that there are three clinical trials are done on Ashwa 30. The first clinical trial was done on stress. The second is on endurance and fatigue, and the third is again on stress. And this is expected to be completed, and the results are expected by October 2025. The first trial on stress is a randomized double-blind placebo-controlled trial to evaluate the effects of Ashwa 30 on cortisol levels. It has two groups. The first group is the Ashwa 30 group, and the second group is the placebo group. 20 volunteers in each arm, and they took Ashwa 30 for 7 days. And on the seventh day, an acute stress mask was given to these individuals for the stress induction, and their cortisol levels were looked into. So this is in brief the MAST protocol. We can see here that MAST is a combination of both the physical stress as well as the mental test. So it is a combination of both physical stressor as well as the mental stressor, which increases the cortisol levels. So here, if we see post-MAST, the salivary cortisol levels in Ashwa 30 group were pretty much low, significantly low when compared to the placebo, which clearly indicates that when Ashwa 30 was taken for 7 days, and the individuals who are exposed to acute stress will be able to deal with the stress in a better manner. That is indicated from their lower cortisol levels. Moving on to the second study, this is again a randomized double-blind placebo-controlled trial on cardiorespiratory endurance and fatigue. In this study, healthy subjects doing regular exercise were recruited. 63 subjects participated in the study, and of course, 30 milligram was given to these individuals for 8 weeks for endurance and fatigue. And the methods used were modified Bruce protocol treadmill test and 20-meter shuttle run. The safety assessment was done. Adverse events, hematology, and biochemical tests were used. So the results go like this. These are the methods used in the study. The gold standard evaluation is VO2 max for any of the cardiovascular endurance. So VO2 max was studied in this study, and VO2 max was studied in the MBPTT. And heart rate, of course, lactic acid levels, and the blood creatinine phosphokinase were studied during the MBPTT. And 20-meter shuttle run also was used for an exertion. And the rate of exertion as well as the fatigue levels were studied in both MBPTT as well as the 20-meter shuttle run test. In addition, safety was also looked into. Adverse events, hematological tests as well as the biochemical tests were done. The modified Bruce protocol treadmill test goes like this. There are 8 stages in the modified Bruce protocol treadmill test. Each stage is for 3 minutes, and each stage there is an increase in the speed and increase in the inclination that puts an exertion on the subjects. And as the speed and inclination increases, there will be difficulty for the subject to continue his exercise on the treadmill. However, after after 8 weeks of ingestion of Ashwa 30, 62% of the people in the Ashwa 30 group took the test, took the treadmill test till stage 8, while only 10% of the people in the placebo could go to stage 8. This clearly indicates that Ashwa 30 intake helps you in taking the exercise for a longer duration. So the endurance benefits are better with Ashwa 30 when compared to placebo. This is the VO2 max. As we know that VO2 max is a gold standard for the endurance benefits. So we can see here that 10% improvement in the VO2 max was observed in the Ashwa 30 group, which was statistically significant when compared to the placebo group. The heart rate max, which is the maximal heart rate, is also significantly increased in the Ashwa 30 group when compared to placebo group, and this increase is around 8%. So both the oxygen consumption, the maximal oxygen consumption, and the maximal heart rates have improved with Ashwa 30 intake. This could be due to the cellular functions, the impact on the cellular functions like the mitochondrial biogenesis. This is again something interesting to note. At the stage 6, when the lactic acid levels were tested during the Bruce protocol treadmill test, the lactic acid buildup in Ashwa 30 was much lesser when compared to placebo group, which clearly indicates that the body shifting from aerobic to anaerobic is slow in the Ashwa 30 group, while it is faster in the placebo group. And when the body is shifting slowly, it helps in better endurance, and it also helps in longer duration of intake of the exercise. So Ashwa 30 helps in less lactic acid accumulation. Blood creatinine phosphokinase is an indicator of the muscle stress. So muscle stress is significantly reduced in the Ashwa 30 group when compared to the placebo group. This also indicates that the muscle stress is significantly reduced by Ashwa 30. Now coming to these parameters on fatigue as well as the exertion. So the fatigue levels, as we see in placebo group, the fatigue levels increased after week 8, and the fatigue levels are decreased in Ashwa 30 group after 8 weeks of intake of Ashwa 30. Similarly, in the shuttle run also, the fatigue levels are increased and the fatigue levels are decreased in the Ashwa 30 group from baseline to week 8. In addition, the exertion, as we see here, the exertion levels perceived by the subjects are not changed in the placebo group, while there is a significant reduction in the exertion levels in the Ashwa 30 group. Similarly, there is a decrease in the exertion of individuals when compared in the Ashwa 30 group when compared to the placebo group. So both fatigue as well as the exertion levels were reduced in Ashwa 30 group when compared to the placebo group. So that gives insights on how Ashwa 30 is useful for stress as well as endurance. It is able to reduce the cortisol significantly within 7 days of intake of Ashwa 30, and it is also able to improve the endurance and also reduce the fatigue levels in Ashwa 30 Ashwa 30 ingested individuals. Coming to the safety, safety was also assessed as a part of this study, in addition to the endurance benefits. The AST, ALT, LDH, glucose, BUN, creatinine, cholesterol, and triglycerides levels were within the normal limits post-ingestion of Ashwa 30. There were no adverse events that were attributed to Ashwa 30 intake, and the LD50 levels are greater than 5 gram, which indicates high safety margin. No genotoxicity was observed in the AIMS test. And in addition to this preliminary safety studies, a full battery of toxicological studies are underway in the GLP lab. So we have come to the end of the presentation that the smallest dose, the smartest science, and the strictly route that was used, and the compliance, cultivated routes also for sustainability, and the high level of safety profile and benefits for endurance and safety at such a low dose helps the formulators to have Ashwa 30 in their formulation. And that's all about Ashwa 30. Thank you for your time.
All right, so excellent. I have to say, as a runner with a race coming up in December, I'm all over this. I need to be taking it starting yesterday. Thank you for all of that terrific science. We're also joined by Aiden from AIDP to field any questions that come in. So please put your questions into the Q&A box. We'll work through those now. First question is, what unique product formats or positioning strategies does Ashwa 30 open up for brands that haven't been possible with traditional higher dose extracts?
So because you have a smaller dosage that you can put into formulations, it really opens up a lot more potential avenues for commercializations. Things that come to mind would be like gummies. With only 30 milligrams a dose, you really have more space to put it into formulas.
Excellent. All right. Another question here on the dose. How do you see low dose, high impact innovation influencing the way formulators and brands approach just more broadly the adaptogen category?
Can you repeat the question, Maggie?
How do you see low dose, high impact innovation influencing the way formulators and brands approach adaptogens more broadly?
So I think specifically the really interesting aspect of Ashwa 30 is that 30 milligram dosage. It kind of shifts away from this hero ingredient where you can actually have a lot more ingredients within a specific commercialization. So instead of having a lot more of your product taken up, usually with ashwagandha, you're seeing around 300 to 600 milligrams of the clinical dosage that is needed. And now we're looking at around a 10th or a 20th of that clinical dose. So it really opens up the overall space to be able to include more ingredients when you go to market.
Excellent. All right. We have a question about the scientific findings. What are the key findings that most clearly demonstrate the ingredient's efficacy at a low dose?
So the key findings include a better endurance and anti-stress benefits, which are evident from the reduced cortisol levels. And of course, the endurance benefits are very clearly evident from the lactic acid buildup reduction, along with the VO2 max increase. So these are some of the very key benefits of this low dose formulation.
All right, excellent. And as we mentioned earlier, the presentations will be available on demand. So anybody who wants to dive into those slides a little bit more closely and look at the data can do that. All right, any final takeaways for us before we move on to our next session?
No, not necessarily. Thank you, Dr. Bharathi.
All
Thank
right.
you.